Now we are going to take into account how is enzyme action inhibited. We had discussed earlier that what are the factors which actually affect the enzyme activity, that either they would be responsible for enhancing the enzyme activity or they would be responsible for declining it. So basically we are going to see how enzyme activity is affected when a chemical which has the tendency to act accordingly and antagonistically to the enzyme would be present, and how the enzyme acts in its presence. Okay. Now when we talk about inhibition of enzyme action, I am talking about that the enzyme activity would be declined to zero, that whatever reaction you were expecting that it has to take place at a very speedy rate, it would be put to a standstill due to the presence of some or other chemical compound which would be in proximity to that particular enzyme. So we have three types of inhibitions which are seen in an enzyme action. First one is competitive inhibition, second one is non-competitive inhibition, and then the other one which is continuously in action in our cells is feedback inhibition. Now please pay attention, apart from these three types of inhibition, the one which is predominantly responsible for bringing the enzyme action to a condition where the enzyme is not working at all, it is denaturation. That is the most important thing. So do remember that the denaturation of an enzyme is, as the name suggests, the enzyme would be either breaking down, there would be a breaking of bonds of the enzyme, the protein would lose its structure, the active sites would be gone. That is what is known as denaturation, and the factors which are responsible for bringing about this denaturation is mostly pH and temperature. Alright, now let's talk about the three types of inhibition apart from denaturation which are responsible for slowing down the enzyme action or stopping the enzyme action. Inhibition means to stop something. So the first one is competitive inhibition. As the name suggests, it is competitive and inhibition would bring some sort of stoppage. Now when I say that competitive, that means, you know, enzyme activity is basically what? The active site is going to be susceptible to the substrate would come and join with it. All that lies in the crux of this entire action is that the active site resembles the structure of the substrate. That is why the substrate is able to come and join and form the ES complex. Over here, the competitive inhibitor would have the same structure as that of the substrate. Now supposedly, consider that I am having this enzyme, it is having this active site, this is the active site for the enzyme. Now this is my substrate that has a shape of this sort. Okay, this is my substrate, this is the binding site, active site as we call it. I am going to represent it as active site. This is my substrate. Now the inhibitor by any chance would have the same structure as that of substrate, at least it would be able to fit into the active site. That means it is having almost a similar structure as that of the substrate. So what happens? It provides a competition for the active site. This is substrate and this is competitive inhibitor. Okay. Now this is what is to be remembered, that both are fighting for a same site because their structure is having a close resemblance. Now this is like a competition, you are fighting for a particular medal and the medal is won, so both would present an equal amount of competition. Same is in the case of this substrate and the inhibitor. The inhibitor would go and combine with the active site and this substrate would be left without an active site, and how can you expect the particular reaction to take place with a lower activation energy if this particular site is already occupied by the inhibitor? So this is what happens in the competitive inhibition. The example that we do is that of, let's consider this enzyme is succinic dehydrogenase. You remember this is an enzyme that is very much important in Krebs cycle, over where succinic acid has to come and combine. Now what happens? The substrate would be succinate. Okay, but succinate has a close resemblance with malonate. So this is what would happen that succinate has to come and combine with the succinic dehydrogenase, but malonate comes and combines with the enzyme, and the entire action, the step which was to be favored by chemical kinetics as that done by these enzymes, that would not be possible because this enzyme has been utilized by a non-productive particular substrate. This was not supposed to be a substrate. The entire process is futile now. Now this enzyme action has been stopped. So this malonate has been able to bring about inhibition of the enzyme action of succinic dehydrogenase. So this is all about competitive inhibition. Now there is another thing. You must have noticed that whenever you are having some sort of infection or allergy, you are given antibiotics which have sulfur drugs in them. Now these sulfur drugs basically, they also work on the principle of competitive inhibition only. Just for your knowledge purpose, you must remember it that what happens in the case of competitive inhibition done by sulfur drugs is that they competitively inhibit the folic acid synthesis in the bacterial cells. Okay, the folic acid synthesis which was to take place inside the bacterial cell, that is inhibited by these sulfur drugs because they are similar in structure as to p-aminobenzoic acid. Please remember, the sulfur drugs, they provide competitive inhibition to p-aminobenzoic acid, which is important for folic acid synthesis in a bacteria. And when this does not take place, the bacteria is forced to meet the fate of death. Alright, so that is how the sulfur drugs protect us from the infections. Now coming to the non-competitive inhibition, as you know, when we simply represent an enzyme, we say that let's take one active site. But because an enzyme is a tertiary structure, it might have many active sites, not just one. So let's consider it this way. This is an enzyme. It is having active site number 1, 2 and 3. Now considering our substrate was to combine at site 1, but we have a substrate or an inhibitor which has more affinity for the site number 2, and it comes and binds over here. So this was to bind at site 1, but because the site 2 had more affinity towards this inhibitor, now this inhibitor would come and combine at the site 2, and when it combines, what happens is that there is complete alteration in the enzyme structure, due to which this combination does not take place. Okay. So this was to combine at site 1, but because some inhibitor has already combined at another site which brings alteration in the structure of the enzyme, this substrate that was to combine, that we wanted to combine, that does not combine, and that particular thing is known as non-competitive inhibition. Because over here the inhibitor did not resemble the substrate first of all, secondly it did not fight for the same site, it came and joined at some other site and it altered the enzyme structure, due to which the combining of substrate was not possible because the active site structure changed. Now as you can see, it cannot combine over here. This is known as non-competitive inhibition, and the biggest example is cyanide poisoning. Now people die of consuming cyanide. Why do they die? Because this cyanide, it goes and provides non-competitive inhibition against the enzyme cytochrome oxidase. Now remember, cytochrome oxidase was the enzyme which was very important in electron transport system. Okay, ETS we did over where the ATP formation was taking place. So it is respiratory inhibitor basically, and it kills the person within seconds. So that is why the cyanide is providing non-competitive inhibition. It does not need to resemble the active site even. It is having lot of affinity for the particular enzyme, that is cytochrome oxidase. It goes and inhibits its activity. Okay. So basically the poisoning takes place by non-competitive inhibition. So there are two types of inhibitions, one is competitive, other is non-competitive. And what about stopping the activity of enzyme. You know, enzyme would come into the scene. We had discussed that if the substrate concentration goes beyond a level, then the activity declines. How is the activity of enzyme regulated by the body? These are inhibitors. There was a denaturation condition. There were factors which were affecting the enzyme activity. What about regulating the enzyme activity? That at this time the enzyme has done what was required of it. It has to stop now. That is done by feedback inhibition, okay, or the allosteric inhibition. Various cells take part in these inhibition processes. They provide a feedback. That means the outcome which was required, the metabolic pathway, there would be some low molecular weight compound and it would go and combine with the enzyme. So we take the example of hexokinase. You remember where hexokinase was required? It was required in the process of glycolysis. Now consider this as hexokinase. Hexokinase converts glucose to glucose 6-phosphate. We have discussed this in glycolysis, respiratory pathway. If not, you can see what was there in the aerobic breakdown of a glucose molecule. Glucose gets converted to glucose 6-phosphate in the presence of hexokinase. Now, how does this hexokinase stop from further breakdown? This low molecular weight, glucose 6-phosphate, would be produced in the presence of hexokinase, and this glucose 6-phosphate would itself come and combine at the active site. That means if the glucose was to come and combine over here, because glucose 6-phosphate, as you know what happens in feedback, that this particular thing has been made enough, now you need to stop the production of that particular thing. This is what happens in feedback. Supposedly you are producing some product, the feedback comes that now it has been taken positively. Now you would stop its particular production. It has been enough. So that is what is the feedback is being provided. Same way, the feedback inhibition or the allosteric inhibition, it would be that glucose was to combine with hexokinase and it would give rise to glucose 6-phosphate. But now, just in order to stop hexokinase from further breakdown of glucose, this glucose 6-phosphate would come and combine with this particular enzyme somewhere over here, and it would alter the activity of hexokinase and no more glucose would be able to combine with it. Now this would act as a feedback to the entire metabolic process, that now hexokinase has to stop its activity. Okay, so this is the feedback inhibition. This in a way keeps on acting inside the cells, because you have to make sure that the enzyme which is working, that stops at a particular stage. Now its production or its working has to be stopped apart from the effect of substrate concentration. So I would repeat it because this is little bit typical, that in the feedback inhibition, some low molecular weight compound would come and stop the activity of the given enzyme. Okay, and that low molecular compound would be a result of the metabolic process itself, which is taking place in presence of that enzyme. Like I gave you the example of glycolysis, the glucose breaks down to glucose 6-phosphate in the presence of hexokinase only, but later on this glucose 6-phosphate goes and combines with hexokinase in order to stop its activity so that no further glucose breakdown takes place. Alright, so we have 3 types of inhibition. This is all about how a presence of a particular chemical compound can stop the enzyme activity. Thank you.